Before we get into our discussion of how we measure the brain, let's quickly step back and take a look at action potentials. And before we do that, let's actually look at how what I mean when I talk about electrical signals. Um, so really quick, let's go over the parts of the brain that are of interest here. So we have oops, our dendrites, and these are what receive inputs from other neurons. And the input in this case is the electrical signal. This is the axon, which carries the output from this neuron, and this is the axon terminal, which sends that output to other neurons. And I've been saying that these neurons, they send an electrical signal, but I haven't actually explained what that means. And when I say it's an electrical signal, I'm talking about a change in the membrane potential. And now you're probably asking, what does that mean, right? So the membrane potential is an electrical charge difference between the inside of a neuron, so what's in here, and the outside, so the extracellular fluid. And now, why is there a membrane potential? The reason for that is neurons are specialized for creating this electrical charge and sending these electrical signals. So neurons have specialized channels and pumps here. Um, so these pink things I'm drawing here could be ion channels, and they go all throughout the cell. Um, and then we could have in green here, oops, not green. In green, could have these ion pumps, which are active diffusers, so they require energy. Whereas the ion channels are passive diffusers and only require a concentration gradient. So the neuron's membrane controls the movement of charged ions. Uh, and in this case, it's mainly sodium and potassium. And because more positive ions are kept outside the cell at rest, when it's not doing anything, no electrical signal, nothing, is negatively charged. And it's found that the charge of that cell at rest is negative 70 millivolts. So the signal here, this is the fun part, is any change in this resting state membrane potential. So it's these changes in the membrane potential that create an action potential. So you might have heard that an action potential is an electrical signal that occurs in a neuron that is transmitted. Another way of saying that is that it's a change in the membrane potential of the neuron that creates the action potential. A lot of potentials here. Don't get them confused. But now we're ready for action potentials. So what are action potentials? I kind of prefaced it there by saying there are electrical signals, and it's exactly that. All it is it's just a change in the membrane potential. So there's a change in the ion concentrations inside and outside the neuron that lead to this electrical signal, as we call it. But it's just, remember, it's just ion concentrations. So when we have sodium here, when they move into the cell, the overall polarity of the cell decreases because you're adding charge to it, adding positive charge. So let's look at what happens during action potential. So what's different from an action potential versus any other type of electrical stimulus is that the the voltage of the cell has to overcome a threshold. A classic example is that you have a leaky bucket and water is being put into the bucket, but it's leaking out the bottom. And an action potential is what would happen if the water overflows the bucket. And the threshold is the leaking. On this graph, let's have the y-axis be millivolts and the x-axis be milliseconds. And you'll note that these are both really small magnitudes of values, and that's because we're working on a really small scale, both time and space-wise. So what occurs during an action potential is that the neuron this could happen a multitude of ways, but essentially the neuron is depolarized to a significant extent. Now, what does that mean? The ion concentration inside the cell becomes less negative in relation to the surroundings. Hence, you depolarize, so you make it less polar, less negatively charged. Um, and what we have found by looking at neuron and measuring these signals is that an action potential only occurs after it becomes depolarized above negative 55 millivolts. So essentially, let's say we have this threshold here of negative 55, and we have our resting state neuron at negative 70. So let's say that there is an external stimulus that causes the neuron um, to receive an electrical signal that via ion channels. So we would see that it would become depolarized. However, unless it comes above this negative 55 millivolts, it's not going to have an action potential. So it could go up here, but then it just depolarizes right back down. And that's because of these red ion-gated channels that maintain the ion concentration gradient. Um, However, now here's the fun part. If this neuron does get 255, negative 55 millivolts, it's going to shoot up, right? The millivolts just going to spike, which is also what we call it. And then it's going to go back down. However, it's not going to go back down to 70. It's going to go back down a little a little under 70 and overshoot. And then it's going to come back down to 70. So this is the action potential. That's it. But let's look a little closer into that. So what we have here are a couple distinct stages in the action potential. We have our relatively resting state neuron right here. Now let's do this in a different color. Let's do blue. And then we have this rapid period of depolarization right here. Which is then followed by, since the polarity is getting larger, it's getting more negative, we'd call it hyperpolarization. Let's do, I don't know what color that is, but do that. And then there's some more um, depolarization here. So let's break it down, right? When this threshold of negative 55 is reached, we have voltage-gated sodium channels opening, which cause sodium to rush into the cell, which rapidly decrease the polarity of the cell. And before that, I just want to mention that in the cell here, um, you're going to have a higher concentration of sodium ions outside and a higher concentration of potassium. Can't draw on that, but a higher concentration of potassium on the inside. It's on the inside, but I can't draw where it's just know that it's on the inside. I'll even say it's on the inside. So now, once the sodium channels open to a certain extent, we also have potassium channels which start to open right around here. And when this happens, the potassium inside the cell rushes outside to maintain that concentration gradient. And this is what causes the hyperpolarization, because you have a rush of positively charged ions outside the cell, which in turn causes the polarity to become more negative. However, what ends up happening here is that the potassium that leaves the cell is much greater than the initial amount in the cell, which is what leads to the hyperpolarization right here. And when this happens, there is this pump which it's called a sodium potassium pump, which pumps sodium out of the cell and potassium in the cell, eventually restoring the balance in the negative 70 millivolts at resting state. And because the membrane potential is changing so much when the action potential starts and even after, once it's repolarizing, this period right here is known as the refractory period, as the neuron is unable to have another action potential during this time. So you'll never end up seeing a neuron that has two um, summed up action potentials, or like a two back to back. So you'll never see two humps like that. Action potentials are a crucial part of the brain. They allow for communication of information, and they allow a lot of the flexing of muscles and the perceiving from our senses. So with this in mind, let's move on to how do we measure action potentials?